A fragment of bone holds a story no history book can tell. Ancient DNA is our closest thing to a time machine, and its revelations are rewriting everything we thought we knew about humanity. By the end of this journey, you will see our entire past through a startling new lens. We will uncover how a vanished human cousin lives on inside you, and why a forgotten population in Siberia holds the key to the first Americans. These are not just scientific facts, they are profound shifts in our self-understanding. Each discovery peels back a layer, revealing a world far more connected and complex than we ever imagined. What does it truly mean to be the only human species left on Earth? That fragile molecule, a whisper of genetic code from the distant past, was once considered impossible to read. For decades, the very idea of recovering DNA from ancient remains was met with profound doubt within the scientific community. The material was too degraded, too contaminated by millennia of exposure, or simply too broken to piece back together. Early pioneers in this field faced an uphill battle, not just against time and decay, but against the prevailing belief that their quest was a scientific fantasy. They worked with painstaking care, often in sterile clean rooms that resembled laboratories for spacecraft, all to prevent a single speck of modern DNA from corrupting their priceless samples. The tools they had were blunt instruments capable of reading only the smallest fragments, but then a technological revolution quietly unfolded. A method called next-generation sequencing changed everything. Imagine trying to read a library that has been burned, its pages scattered to the wind. Old technology could only pick up a few charred sentences at a time. Next-generation sequencing allowed scientists to gather every single ash, every tiny fragment of letter, and use immense computing power to reconstruct the original texts all at once. This was the key that unlocked the vault. We were no longer just studying bones as silent objects. We began reading them as biological diaries. This shift is monumental. It means we can now ask questions that archaeologists could only dream of. We can trace not just where people traveled, but who they loved and raised a family with. We can see the shadow of ancient illnesses in their genes and understand what they may have endured. We can even detect what they ate from the microscopic clues left behind. This is the new lens through which we view human history, moving beyond the stone tool and the pottery shard to understand the heartbeat of the people who made them. And the very first entry in this new diary introduced us to a relative we never knew we had, a ghost that had been hiding in our family tree all along. That first ghost in our genome had a name, Neanderthal. The comparison was stark. On one side, the genetic blueprint of a modern person. On the other, the painstakingly reconstructed code from a fragment of bone tens of thousands of years old. The data revealed a truth that rewrote the human story in a single stroke. People of European and Asian descent today carry a small but significant piece of Neanderthal within them, between 1% and 4% of their entire genetic makeup. This was not a case of distant cousins evolving on separate paths. This was evidence of something profoundly intimate. When our own Homo sapiens ancestors ventured out of Africa, they did not simply replace the other humans they encountered. In many places, they met them, lived alongside them, and built families with them. Neanderthals were not a dead end. They became part of us. This genetic handshake is far from silent. That ancient inheritance influences real, tangible aspects of who we are today. It shapes how some of our immune systems recognize and fight illness, a gift from a cousin who had long adapted to the environments of Eurasia. It can affect traits like hair texture and skin tone, and even plays a role in our susceptibility to certain modern health challenges. The old cartoon image of the grunting, primitive caveman vanished forever. In its place stood a portrait of a resilient, intelligent, and adaptable people, so similar to us that when we met, the differences sometimes melted away. Their legacy is not in museums but in the living biology of billions of people. This discovery did more than add a branch to our family tree. It revealed that the tree was woven together. And it raised a thrilling question. If we carried traces of one lost cousin, could there be another? The search led scientists to a single, tiny finger bone found in a remote Siberian cave, a bone that would point to a ghost population no one had ever dared to imagine. That single finger bone from a Siberian cave held a secret that would dwarf the Neanderthal revelation. It was not just another ancient human, it was a ghost, a population so distinct that its very existence had been erased from the fossil record. Scientists named them the Denisovans after the cave where the bone was found, but they are known almost entirely from their genetic echo. This was a monumental shift, the first time an entirely new branch of the human family tree was discovered not by unearthing a skull, but by decoding a genome. It proved the prehistoric world was a crowded stage, populated by multiple forms of humanity whose stories are only now coming to light. The real shock, however, was not just that they existed, but where their legacy endured. When researchers compared the Denisovan genome to modern populations, they found a stunning pattern. People from Melanesia and Aboriginal Australian communities carried significant portions of Denisovan DNA, in some cases even more than the Neanderthal inheritance found in Europeans. This was the first concrete biological evidence of an epic, ancient journey. It painted a picture of early modern humans moving through Asia tens of thousands of years ago, encountering and mixing with these mysterious Denisovans. That genetic signature carried across generations and across the vast ocean to reach islands like New Guinea and Australia, became a living map of a migration that left almost no physical trace. The Denisovan story is a humbling lesson in how much of history is lost, yet how fragments of it are meticulously preserved within us, waiting for the right key to unlock their story. It showed that DNA could trace not just family lines, but entire continental sagas written in the language of biology. And if genetics could map such a vast journey across Asia and into the Pacific, what could it reveal about the very first footsteps into an entirely new world, a land where no human had ever lived before? The journey into the Americas stands as one of humanity's final epic voyages into a true wilderness. For generations, the story of that first crossing was a puzzle with missing pieces, debated by archaeologists who studied stone tools and ancient campfires. Some argued for an early coastal route, others for a later inland passage after the glaciers retreated. The theories were compelling, but they were built on fragments. Then, a child's resting place in Alaska offered a new kind of clue, written not in artifacts but in ancestry. The DNA from this individual revealed a population scientists called the ancient Beringians. Their genetic story was a revelation. It showed that the ancestors of all indigenous peoples in the Americas did not enter the continent in one single, swift movement. Instead, they spent millennia isolated in a land bridge region called Beringia, a vast, now submerged landscape that once connected Asia and North America. Roughly 20,000 years ago, while still in this northern holding area, a group split off. This was the ancient Beringian branch. The rest of the population would later move south, giving rise to all other indigenous lineages across two continents. This genetic evidence provided a clear, slow-motion timeline for the peopling of an entire world. Later discoveries, like the genome from a young child buried with great care in Montana over 12,000 years ago, confirmed this unified origin story. That individual, known as the Anzac child, carried DNA that connected directly to contemporary Native American communities. It was a powerful, living link across an immense span of time. These genomes together drew a new map, They showed a founding population moving southward, then branching into the incredible diversity of cultures that would flourish from the Arctic to Tierra del Fuego. The long-standing controversies about how many migrations occurred or who came first were settled by this biological roadmap. It told a story not of separate arrivals, but of one shared ancestral journey, a testament to the resilience and adaptability of those first families who looked upon an untouched continent and called it home. This genetic clarity did more than answer old questions. It reframed our entire understanding of this last great human expansion, revealing a cohesive narrative written in the bones of children, waiting patiently for thousands of years to be heard. And if DNA could so clearly chart a migration across continents, what could it reveal about the stability of the societies those migrants built? Could it detect the moment a thriving civilization began to quietly unravel from within? The quiet stability of those first American communities paints a picture of deep-rooted continuity, but our history is also shaped by moments of profound and sudden change. One such moment reshaped the entire world, a revolution so fundamental it altered our relationship with the land itself. We call it the dawn of agriculture, the Neolithic Revolution. For a long time, archaeologists debated how this new way of life, with its domesticated crops and settled villages, spread across continents like Europe. Was it simply a brilliant idea passed from one group of hunter-gatherers to another like a new tool-making technique? Or was it something more? The answer lay not in the seeds or the stone sickles, but in the bones of the very first farmers. When scientists finally read the ancient DNA from those early agricultural communities in Europe, they were met with a genetic story that rewrote the narrative entirely. These pioneering farmers were not local hunter-gatherers who had decided to plant a few seeds. They were newcomers, genetically distinct, with ancestry tracing directly back to populations in Anatolia, in modern-day Turkey. Around 8,000 years ago, a wave of migration began. These farming communities moved generation by generation, bringing their wheat, their sheep, and their entire way of life with them into new lands. The genetic data reveals this was not a gentle blending of ideas, but a powerful demographic movement. In many regions, the existing hunter-gatherer populations were largely replaced over time. Scientists call this process demic diffusion, meaning the spread of a new culture was carried literally in the bodies and the families of the people who practiced it. This discovery fundamentally changed how we understand cultural transformation. It proved that the most revolutionary ideas in human history are sometimes inseparable from the people who carry them. The Neolithic package was not just a set of instructions, it was a complete population with their own stories, their own ancestry, and their own genetic legacy that would become the bedrock of much of modern Europe. This genetic upheaval set a powerful precedent, showing that our species' story is not one of peaceful, static existence, but of dynamic movements that reshape continents. And if the peaceful adoption of farming could involve such a dramatic population shift, what kind of genetic story would accompany a far more turbulent chapter, one written not by plows but by conflict and conquest? The transition from farming villages to a world of warriors and wheels arrived on horseback from the eastern grasslands. A monumental study of genomes from the Bronze Age revealed a genetic shockwave that rippled across the ancient world. Roughly 5,000 years ago, a wave of people from the vast Pontic Caspian steppe, a region north of the Black Sea, began a movement that would reshape Europe and parts of Asia. These were the Yamnaya, a culture defined by their mastery of the horse, their use of wheeled carts, and a pastoral way of life centered on herds. Their genetic signature, once identified, appeared with stunning force in the DNA of individuals buried centuries later in Central and Northern Europe. In some regions, they replaced nearly 75% of the existing ancestral population. This was not a slow blending, but a powerful demographic tide. The implications of this genetic upheaval reached far beyond biology, touching the very words we speak. For centuries, linguists had puzzled over a profound mystery. Languages as diverse as English, Spanish, Greek, Persian, and Hindi share deep, fundamental roots, suggesting a common ancestral tongue scholars call Proto-Indo-European. But where did it originate and how did it spread so widely? The archaeological record offered competing theories, but the genetic data provided a startlingly clear answer. The timing and the path of the Yamnaya migration matched the proposed spread of Indo-European languages perfectly. Here was the likely vehicle, a highly mobile, horse-riding culture whose families and communities carried their language across continents as they moved. Their legacy is not just in ancient burial mounds, but in the living speech of billions. When you say the words mother or night in English, you are echoing a linguistic thread that these steppe pastoralists carried westward thousands of years ago. This discovery solved a centuries-old puzzle by showing how culture, language, and genes can be inseparable travelers. The Yamnaya did not just bring new ideas, they brought themselves, their families, and their entire way of life. Their success created a biological and cultural layer that underlies much of modern Europe, a testament to how mobility has always been a powerful engine of human change. It reveals a world where the open steppe was not a barrier but a highway, connecting distant peoples in ways that permanently altered the course of history. If the movement of people could so dramatically reshape languages and lineages across Europe, it makes us wonder what other, quieter forms of connection existed. What stories could be told by the objects that moved between cultures, not with conquering armies, but through the hands of traders along silent, glittering paths? The movement of people and goods along ancient trade routes did more than spread ideas and wealth. It carried unseen passengers. Pathogens have been a silent, powerful force in human history, their impact often invisible in the archaeological record until a single technological breakthrough. Scientists discovered they could extract not just human DNA from ancient teeth, but also the genetic remnants of the microbes those individuals carried. This opened an entirely new archive, a molecular record of ancient illness. What they found rewrote our understanding of when great plagues began to shape our world. The bacterium responsible for the Black Death, Yersinia pestis, was not a medieval invention. Its genetic signature was identified in skeletons from the Bronze Age, dating back over 5,000 years. This was a staggering revelation. It meant that devastating epidemics capable of reshaping societies were a recurring threat long before historians had any written accounts of them. These ancient versions of the plague may have been less transmissible than the strain that swept through 14th century Europe, but for the small, interconnected communities of the ancient world, their impact could have been catastrophic. This discovery offers a compelling explanation for some of prehistory's great mysteries: the sudden decline of large Neolithic settlements, or the rapid abandonment of certain regions, might not have been due to climate change or conflict alone. A swift and terrible outbreak of illness could have shattered a community's social fabric, leaving survivors to disperse and seek new homes. Disease became a new character in our historical narrative, an evolutionary pressure as real as any predator or drought. By tracking the genetic changes in these ancient pathogens alongside shifts in human populations, scientists can now see a chilling correlation. Times of great human migration and upheaval often coincide with evidence of novel, virulent diseases. The microbes evolved as we did, and our own genomes bear the scars of that ancient arms race. Certain genetic variants that offer resistance to diseases like the plague are found at high frequencies in populations descended from those who survived these early outbreaks. It is a permanent mark of resilience written into our biology. This reminds us that history is not just the story of kings and conquests, but also of the microscopic world. Our fate has always been intertwined with the viruses and bacteria we carry, a delicate balance that has at times tipped entire civilizations toward an uncertain future. And if invisible microbes could leave such a profound mark on our past, what other unseen forces were at play? What happens when we look beyond the biology of humans and pathogens to the very environment that sustained them, to the plants and animals that made survival possible? The story of humanity is not confined to continents, but written across the scattered islands of the world. For a long time, the rich archaeological tapestry of the Caribbean, with its distinct pottery styles and ceremonial sites, suggested a single, unfolding story of cultural development. The narrative seemed straightforward. The first people arrived, they thrived and evolved their traditions over thousands of years, eventually giving rise to the complex Taino societies encountered by European explorers. But ancient DNA from the islands told a different, far more dramatic tale. The genetic evidence revealed a history not of continuity, but of profound replacement. The Caribbean was settled in two distinct, sweeping waves. The first inhabitants were archaic foraging groups who likely island hopped from Central or South America over 6,000 years ago. They lived on the islands for millennia, leaving behind traces of their presence in scattered stone tools. Then, roughly 2,500 years ago, a second wave arrived. These were ceramic-making farmers who sailed from the northeast coast of South America. They brought a new way of life, new technologies, and a very different genetic signature. The analysis showed a staggering genetic turnover. The descendants of those first archaic settlers were almost completely replaced by the incoming farming communities. This was not a peaceful blending of cultures, but a demographic reset across the entire archipelago. This discovery overturned decades of archaeological theory. The changing pottery styles were not simply an evolution of local tradition. They were the material footprint of entirely new people. It highlighted how island worlds, often seen as isolated laboratories of culture, can be genetic crucibles where history moves in sudden, powerful currents. The sea was not a barrier but a dynamic highway for migration, capable of delivering entirely new chapters of human history to distant shores. This pattern of layered settlement and replacement, first revealed in the Caribbean, would become a recurring theme as scientists turned their genetic lens toward the vast expanse of the Pacific Ocean, where an even more incredible story of navigation and endurance awaited discovery. The vast Pacific presented the ultimate test of human ingenuity, a world of endless blue dotted with specks of land. To settle it required not just boats, but a profound knowledge of stars, winds, and waves passed down through generations. On the easternmost frontier of this Polynesian expansion lies perhaps the most isolated inhabited island on the planet, Rapa Nui, known to the world as Easter Island. Its silent, towering stone faces, the moai, have gazed inland for centuries, their creation spawning countless myths. For a long time, a popular narrative took hold, one of a complex civilization that overreached, collapsed into conflict, and vanished, leaving only these stone giants as a warning. It was a compelling story of ecological folly, but ancient DNA asked a simple, direct question of the bones left behind. Who were these people really? The answer, written in their genes, was unequivocal. The individuals who built the moai carried pure Polynesian ancestry, their genetic lineage tracing directly back to the same source populations that settled Hawaii, New Zealand, and Tahiti. There was no genetic signal from South America, no evidence of contact with the continent before the arrival of European ships. This single finding dismantled decades of popular speculation about pre-Columbian transoceanic voyages to the island. The mystery of the moai was not solved by outsiders, but by the descendants of the greatest navigators in human history. The genetic story grew even more profound when researchers looked at the diversity within the genomes. They found evidence of a severe genetic bottleneck, a telltale sign of a founding population that was incredibly small, perhaps just a few dozen people who made that final, daring voyage into the unknown. For generations, this tiny community survived and thrived in utter isolation, a testament to profound resilience. Their story is not one of mysterious collapse, but of breathtaking adaptation. They adapted to a limited environment, built a unique and awe-inspiring culture, and their legacy is not a cautionary tale of failure, but a monument to human tenacity. The Moai were not built by a lost civilization, but by a living, connected people who chose to honor their ancestors in stone on the farthest edge of their world. Genetics here did more than correct a historical record. It restored a narrative of strength and continuity to a people whose real story had been overshadowed by myth. The vast reach of an empire is measured in more than just miles conquered or taxes collected. It is written in the very ancestry of the people who lived at its heart. Ancient DNA studies from the city of Rome itself have now quantified the incredible human tapestry woven by the Roman Empire. Analysis of individuals buried during the imperial period, from the 1st to the 3rd centuries, reveals a genetic landscape that looks almost like a snapshot of the entire Mediterranean world. The population of the capital was a stunning mix, with significant ancestral contributions from the Near East, North Africa, and across Europe. This was not a subtle blending, but a powerful influx, a direct biological reflection of the empire's machinery of conquest, trade, and slavery. People were moving from every corner of the known world to the metropolis at its center, drawn by opportunity or brought by force, and they left families behind. The genetic data provides a tangible, personal dimension to the historical idea of Rome as a melting pot. You can see the legacy of a soldier from Syria, a merchant from Carthage, and an administrator from Gaul, all contributing to the genetic fabric of the city. But this vibrant diversity was not permanent. The second half of the story is just as revealing. After the political collapse of the Western Empire, the genetic signature of the city shifted dramatically. That broad Mediterranean diversity faded, and European-like ancestry became dominant once more. This ebb and flow is a profound lesson. It shows that empires create fleeting genetic landscapes, powerful demographic waves that reshape a region for centuries, only to recede when the political forces that created them dissolve. Rome's genetic legacy was not about replacing one population with another forever, but about a temporary, intense fusion that left a permanent, if subtle, mark on the historical and biological record. This dynamic process of genetic influx and subsequent contraction is a pattern we can now trace in other imperial contexts, revealing a fundamental rhythm of human history where politics and demography are inextricably linked. A rhythm that would play out again on a different stage, where the clash of cultures was written not just in chronicles, but in the bones of warriors on a forgotten battlefield. The rhythm of empire gave way to a new pulse in Britain. After the last Roman legions departed, the island entered a period so shadowed that historians once called it the Dark Ages. Legends spoke of a king named Arthur holding back the tide, while old chronicles told of fierce invaders from across the sea. For centuries, scholars debated what truly happened. Was this a story of a small warrior elite conquering and ruling over a largely unchanged native population, or something far more profound? The answer lay waiting in ancient cemeteries across England. When researchers analyzed the DNA from individuals buried in the 5th and 6th centuries, the genetic story was stark and decisive. Approximately 75% of the ancestry in early medieval England did not trace back to the Romano-British population that came before. Instead, it arrived in a wave from the coastal regions of modern-day Denmark, northern Germany, and the Netherlands, the homelands of the Angles, Saxons, and Jutes. This was no mere change in rulers. This was a demographic transformation on a scale that rewrites a chapter of history. the data revealed a large-scale migration, likely involving tens of thousands of people, entire families, farmers, craftspeople, and warriors crossing the North Sea to settle a new land. The cultural shift from post-Roman Britain to Anglo-Saxon England was not just a change in fashions or pottery styles adopted by a local populace. It was carried in the very lineage of the people themselves. This genetic influx perfectly aligns with the complete transformation of the language, the place names, and the material culture that defines the period. Old English, with its Germanic roots, did not slowly evolve from the Celtic and Latin spoken before. It arrived with a new population, becoming the foundational tongue of the land. This discovery settled a debate centuries old. The Anglo-Saxon migration was real, substantial, and its impact was written not just in law codes and epic poetry like Beowulf, but in the biological fabric of the nation. It demonstrates with stunning clarity how political and cultural revolutions are often powered by the movement of people, a reminder that the pages of history are turned not only by ideas, but by the families who carry them to new shores. The genetic upheaval of the Anglo-Saxon migration was not the final chapter written in Britain's bones. Centuries later, new sails appeared on the horizon, and their story, too, has been rewritten by ancient DNA. The Vikings have long been etched in history as fierce raiders, their dragon-headed ships a symbol of sudden terror. But genomes from the Viking Age, extracted from burial sites stretching from Scandinavia to the British Isles, reveal a far more nuanced portrait. These Norse explorers were not a monolithic horde of pillagers. They were a complex network of traders, settlers, and diplomats whose greatest export was often themselves. The genetic data shows a clear pattern of movement and settlement, particularly in regions like the Danelaw in eastern England, where Scandinavian ancestry entered the population. But the flow was not one way. In a stunning twist, the study found that some individuals buried with classic Viking artifacts in Scandinavia, like swords and shield bosses, carried genetic ancestry from southern Europe and even Asia. This reveals a hidden dimension of their voyages. The Vikings did not just take treasure, they brought people back. They integrated individuals from distant lands into their communities, whether through the hardship of slavery or through marriage and family bonds. This finding fundamentally redefines what it meant to be a Viking. Their identity was not a rigid genetic club, but a cultural and social role one could adopt. A person with ancestry from the Mediterranean could be buried as a Norse warrior, their story woven into the saga. This paints a picture of the early medieval world as surprisingly mobile and interconnected, where genes flowed as steadily along trade and raid routes as silver and silk. The Viking Age was not a clash of pure, isolated peoples, but a dynamic era of exchange, conflict, and integration, leaving a genetic signature that whispers of a world far more complex than the legends of raiders alone could ever tell. This genetic mobility shows that even in an age remembered for its axes, human connections were being forged across astonishing distances. The genetic mobility of the Viking Age reveals a world of constant exchange, but some exchanges carry a far heavier toll. In the middle of the 14th century, a biological catastrophe swept across continents, one that would etch itself not just into history books, but into the very code of human life. The Black Death was a storm of unimaginable scale, a pandemic that reshaped societies, economies, and the course of history itself. For a long time, its story was told through the chilling statistics of chroniclers and the silent, mass graves uncovered by archaeologists. But now, ancient DNA has allowed us to read a new chapter from that terrible time, written in the bones of its victims and, astonishingly, in the biology of its survivors. By extracting genetic material from individuals buried in plague pits across Europe, scientists could finally see the pandemic not just as a historical event, but as a direct, powerful force of evolution. They asked a profound question: In a population where perhaps half of all people did not survive, what made the difference? The answer lay in subtle variations in the human genome. Researchers identified specific genetic variants that appeared to offer a survival advantage during the outbreak. Think of it as a biological lottery, where holding a certain sequence of letters in your DNA might have slightly improved your odds of resisting the infection. This was natural selection operating with brutal historical clarity. The pandemic was not a random tragedy. It was an evolutionary pressure that filtered the human population in a matter of a few horrific years. Those who carried these protective variants were more likely to live, to have children, and to pass those life-saving genes on to future generations. As a result, the frequency of these variants in European populations increased dramatically in the generations immediately following the plague. The event literally sculpted the genetic landscape of an entire continent. Yet this ancient protection came with a complex modern price. The same immune system genes that helped people survive the plague are now understood to be linked to a higher risk of developing certain autoimmune diseases. In a world without the constant threat of that specific bacterium, an overactive, vigilant immune system can sometimes turn on the body it is meant to defend. This is the profound and haunting legacy of the Black Death. It gifted a generation with resilience, but that gift echoes down the centuries as a potential vulnerability. Our biology carries the scars of past battles, instructions written in crisis that our modern bodies must still interpret. This discovery transforms how we view history and health. It shows that our genome is a living palimpsest, a document where the traumas and triumphs of our ancestors are recorded in biological script. A pandemic from 700 years ago is not a closed chapter. It is an active paragraph in the story of human immunity, reminding us that we are all, in a very real sense, the children of survivors. That deep connection to place, spanning tens of thousands of years, finds its most profound expression far to the south. While Europe's story was one of repeated genetic upheaval and replacement, another narrative of astonishing continuity was quietly unfolding on an isolated continent. Genetic studies of Aboriginal Australians, combined with the precious few fragments of ancient DNA recovered from that land's harsh climate, reveal a lineage of breathtaking depth. Their ancestry traces back roughly 50,000 years, marking them as representatives of one of the oldest continuous civilizations on our planet. This is not a claim based on legend alone, but a fact written in the very cells of living people, a biological chronicle that stretches back to the very first footsteps on that ancient shore. The genetic story here is one of remarkable unity. It shows that the ancestors of all Aboriginal peoples descended from a single, founding population that arrived during that initial wave of human expansion out of Africa. Once they arrived, they thrived in profound isolation. The data reveals minimal genetic input from later migrations until the very recent past. For millennia, this was a story written by one people on their own terms. This genetic evidence provides powerful, scientific support for the deep spiritual connection to country that is central to Aboriginal cultures. That connection is not a metaphor, but a lived reality built over 2,000 generations of continuous care, knowledge, and adaptation. They did not simply inhabit Australia. They learned its every rhythm, from the lush northern rainforests to the central deserts, diversifying into countless distinct nations and languages while maintaining an unbroken biological and cultural thread back to the first explorers. This discovery fundamentally challenges simplistic models of human history that emphasize constant displacement and replacement. Here, resilience took the form of continuity. It stands as a monumental testament to cultural endurance, showing that human success can be measured not only in conquest and migration, but in the profound, sustained relationship between a people and their homeland across an almost unimaginable span of time. Their story rewrites our understanding of civilization itself, proving that its deepest roots are grown not in stone cities, but in enduring knowledge passed faithfully from parent to child across hundreds of centuries. Africa's story was always the deepest, but for the longest time it was the hardest to read. The very cradle of humanity presented a profound paradox. While every person on Earth carries an inheritance that leads back to this continent, the ancient genetic record there seemed frustratingly silent. The reason was environmental. Heat and humidity are the great enemies of DNA preservation, breaking down that fragile molecule over millennia. For years, the early chapters of our species' story in Africa were like a library where the oldest, most valuable books had crumbled to dust, leaving only their later copies on other continents to be studied. We knew the outline, but the intimate details, the movements and meetings of the very first Homo sapiens communities, remained a mystery written in fading ink. Then, a technological breakthrough met a moment of archaeological fortune. Scientists refined their methods to extract genetic material from even the most degraded remains, and they turned their attention to rare, precious finds from places like the highlands of Malawi and the rock shelters of Tanzania. What they recovered was not just a few lines of text, but entire missing chapters. These ancient African genomes revealed a continent in constant, dynamic motion, long before any human ever set foot on another land. They showed multiple, previously unknown population expansions, contractions, and interactions, a complex dance of groups meeting, mixing, and moving apart across hundreds of thousands of years. This discovery fundamentally rewrites the opening narrative of our species. Africa was not a static cradle where humanity evolved in isolation and then simply walked out. It was a vast, ever-changing stage where the first acts of the human story were already full of drama, migration, and connection. The deepest roots of human diversity are not found scattered across the globe, but woven right here in the ancient soil of the continent itself. It emphasizes that the great human migrations we've traced to Europe, Asia, and the Americas were merely continuations of a pattern set in motion in our original homeland. Our restlessness, our ability to adapt and blend, is a trait born in Africa, a legacy written in the very first strands of our shared genetic code. Our own genetic journey is mirrored in the stories of the other great beings who shared our ancient world. The final chapter of the woolly mammoth, a creature that walked alongside our ancestors for millennia, was once a simple tale of climate change and human hunters. But ancient DNA has now chronicled its last days with heartbreaking detail, revealing an ending far more complex and intimate than we ever knew. Isolated on a remote Arctic refuge called Rangel Island, a tiny population of mammoths clung to survival for thousands of years after their mainland cousins had vanished. Cut off from the genetic diversity of a larger species, they were trapped in a biological echo chamber. Generation after generation, they could only breed with their close relatives. The result was a devastating genetic meltdown, a slow accumulation of harmful mutations that no natural selection could purge from such a small group. The decoded mammoth genome tells a story of a species quietly unraveling from within. These last mammoths suffered from a loss of their sense of smell, a critical tool for finding food and sensing danger. Their famous fur became thin and translucent, losing its insulating power against the bitter cold. Their ability to digest food properly was compromised. They were, in essence, a ghost population long before the final individual passed. This genetic catastrophe does not excuse the role of a shifting climate or the arrival of human hunters, but it adds a profound new layer to their extinction. It shows that a species can be biologically doomed long before its last breath, weakened to a point where even a small additional challenge becomes insurmountable. The mammoth story is a stark, sobering lesson written in ancient code. It warns us about the invisible erosion that occurs in any small, isolated population, a slow bleed of genetic vitality that compromises health, resilience, and the very ability to adapt. For conservation biologists today, it is a crucial case study, highlighting that saving a species from the brink requires more than just protecting a few remaining individuals. It requires safeguarding the genetic diversity that is the foundation of all future survival. In a way, the mammoth's fate echoes the quieter endings we have seen in our own human family, a reminder that the story of life on Earth is often written not in dramatic cataclysms, but in the slow, silent accumulation of small, irreversible losses. The silent unraveling of a species finds a strange parallel in the most celebrated and seemingly eternal of human lineages, the pharaohs of ancient Egypt. For centuries, these rulers were known only through their monumental art and sacred texts, depicted as flawless, godlike figures frozen in stone. Ancient DNA has now begun to pull back that golden mask, not to diminish their legacy, but to understand them as people within a living, breathing, and genetically complex world. Extracting genetic material from Egyptian mummies is a formidable challenge, as the embalming process and the relentless desert heat degrade DNA over millennia. Yet, where science has succeeded, it has rewritten the demographic history of the Nile itself. One landmark study focused on individuals from the New Kingdom period, the era of powerful rulers like Rameses the Great. The genetic signature of these ancient Egyptians revealed a startling story. They showed a closer ancestral connection to populations from the Near East, from the Levant and Anatolia, than to the genetic makeup of people living in Egypt today. This single finding illuminates a profound truth about the long arc of history. The population of Egypt has never been static. Over the last 2,000 years, a continuous flow of people from across Africa, later Arab migrations, and other movements have woven new threads into the nation's genetic tapestry, enriching and transforming it. The pharaohs, therefore, represent a specific chapter in a long, dynamic story of human movement in the region, a snapshot of a population that has since evolved through countless subsequent generations. Beyond these broad demographic shifts, genetics offers an intimate look at family dramas within palace walls. By analyzing the DNA of royal mummies, researchers have been able to trace lines of kinship with remarkable precision. They have confirmed the parentage of famous kings, validating historical records etched in temple walls. In other cases, they have uncovered biological relationships that history forgot, revealing unknown connections that hint at political alliances or private choices lost to time. This transforms our understanding from a study of symbolic icons to a recognition of biological individuals, with all the complexities of health, heredity, and family that implies. We see not just a god-king, but a son, a father, or a cousin, navigating the immense weight of dynasty within a genetic landscape that was always in motion. This delicate genetic work reminds us that even the most seemingly immutable civilizations are rivers of humanity, constantly fed by new tributaries, their strength lying in their ability to change and endure across the ages. The silent departure from a great city leaves a different kind of ghost. Across the ocean, on the banks of the Mississippi River, the largest pre-Columbian settlement north of Mexico once thrived. Cahokia was a marvel, a bustling metropolis of perhaps 20,000 people centered around enormous earthen mounds. Then, around the middle of the 14th century, the city emptied. The great plaza fell quiet, and the wooden palisades slowly decayed. For generations, the question haunted archaeologists: what happened? The silence seemed to demand a dramatic answer, a cataclysmic invasion, a conquering force that swept in and destroyed the civilization. The ground itself held clues, but they were ambiguous. To find a clearer answer, researchers turned to the one witness that never forgets its origins: ancient DNA. They analyzed the genetic material from individuals buried at Cahokia both during its peak and in the centuries after its decline. the results were definitive and deeply revealing in their simplicity. There was no genetic evidence of a new population arriving to replace the city's builders. The same ancestral lineages persisted in the region. This single finding rewrote the narrative of Cahokia's end. It ruled out one of history's most common plotlines, the story of violent conquest and population replacement. The people did not vanish, they walked away. This genetic continuity points to a more complex and perhaps more human story of unraveling. Without the shock of an external enemy to blame, historians could focus on the internal pressures that can cause a society to quietly come apart. A prolonged drought might have strained the agricultural system that fed the great city. Political or religious upheaval could have fractured the social glue that held the community together. Perhaps it was a slow accumulation of hardships that made the grand urban experiment unsustainable, leading families to make a collective choice to disperse, to return to smaller, more resilient communities in the surrounding woodlands and river valleys. Their genetic legacy did not end, it simply merged back into the wider tapestry of the region. This use of ancient DNA is a powerful lesson in historical method. Sometimes the most important answer is knowing what did not happen. By eliminating the dramatic invasion scenario, genetics sharpened the focus, allowing archaeologists to investigate the subtle, interwoven threads of environment, society, and belief that truly shape the destiny of a civilization. It reminds us that not all endings are explosions. Some are a long, slow sigh, a decision to leave the grand monuments behind and carry on elsewhere. Herodotus wrote of them with a mix of awe and dread, a people who drank the blood of their enemies and rode like the wind across an endless sea of grass. The Scythians were the ultimate nomads of the ancient steppe, a name that evoked fierce warriors and golden treasures pulled from burial mounds. Yet for all the Greek historians' vivid tales, their true origins remained a shadowy mystery. Were they a single, pure people from the east, a loose collection of tribes? Ancient DNA has now pulled back the curtain on this legendary confederation, revealing a reality far more interesting than the stereotype. The genetic story told by their remains is one of stunning diversity. Across the vast expanse of the Eurasian steppe, from the Black Sea to the Altai Mountains, individuals identified as Scythian by their artifacts carried a wide spectrum of ancestry. Some had deep roots in Eastern Eurasia, others showed strong connections to Western Eurasian populations, and many were a blend of both. This was not a monolithic ethnic group, but a political and cultural identity, a way of life adopted by many different peoples. The Scythian world was a melting pot on horseback, a network united by shared customs, a mastery of mounted warfare, and a mobile economy, not by a single, exclusive bloodline. This genetic tapestry reflects the incredible reach and connectivity of the steppe itself, a highway where ideas, goods, and people flowed as freely as the grass bent under the hooves of their herds. But the revelations did not stop at origins. For centuries, accounts of fierce women among the steppe tribes were dismissed as Greek myth or exaggeration. Then genetics provided definitive proof. In burial mounds across the region, skeletons buried with full sets of weapons, bows, and arrows, honors reserved for elite warriors, were genetically confirmed to be female. These were not symbolic gestures. Analysis of their bones showed the telltale muscle attachments and physical stress markers consistent with life spent riding and training for combat from a young age. Here was irrefutable evidence of women who held martial status and power within their societies, challenging simplistic notions of gender roles in the ancient world. The Scythian legacy, read through their genes, transforms them from a vague, barbaric horde in the historical imagination into a sophisticated and adaptive civilization. Their strength lay in their fluidity, their ability to absorb diverse peoples, to grant status based on skill and role, and to thrive across a landscape that more rigid societies could not control. They remind us that some of history's most powerful empires were not built on stone foundations, but on mobility, inclusion, and the deep, personal bonds of the riding party, leaving a genetic signature as mixed and dynamic as the wind-swept steppe they called home. The precise moment our paths diverged is written in the very letters of our genetic code. By comparing the complete genomes of modern humans, Neanderthals, and Denisovans, scientists have built a tool of astonishing precision, a molecular clock. This clock does not tick with gears, but with mutations, the tiny, random spelling changes that accumulate in DNA at a surprisingly steady rate over vast stretches of time. By counting the genetic differences between us and our ancient cousins, researchers can calculate backwards to the last common ancestor we all shared. The answer it provides is reshaping our understanding of deep time. This genetic evidence suggests our lineage parted ways with the branch that would lead to Neanderthals and Denisovans somewhere between 550,000 and 765,000 years ago. This is a significantly earlier split than some estimates based solely on the fossil record had proposed. That date is more than just a number on a timeline. It redefines a pivotal and crowded chapter in human prehistory known as the Middle Pleistocene. Imagine this period not as a simple, linear march toward modernity, but as a long, experimental phase where multiple forms of humanity evolved in parallel across the Old World. Our ancestors in Africa were on one path, while the ancestors of Neanderthals and Denisovans were on another in Eurasia, each adapting to their own environments and challenges for hundreds of thousands of years before their descendants would ever meet again. This refined timeline provides the crucial temporal framework for everything that followed. It gives us the scale of our separate journeys. It means that by the time the first Homo sapiens eventually walked out of Africa, the Neanderthals they encountered were not close relatives, but cousins separated by over 500,000 years of independent evolution. They had become truly distinct populations, yet not so different that they could not, on occasion, become one family. This genetic clock does more than date a divergence. It measures the depth of our shared history and the incredible endurance of each lineage. It reminds us that the story of humanity is not a short sprint, but a marathon run over epochs where fate occasionally brought long-lost branches of the family tree back together under a shared sky. That molecular clock measures a deep separation, but the story of who we are is also woven from threads of populations we never knew existed. A single genome recovered from a site in northeastern Siberia dating back over 10,000 years told a story of a world far more genetically complex than any map of modern peoples could show. This individual was not simply an ancestor of today's Siberians or a direct forebear of Native Americans. Their genetic signature was something entirely distinct, a unique branch on the human tree that scientists named the ancient North Siberians. They carried a blend of ancestry from both Western and Eastern Eurasian sources, but mixed in proportions seen nowhere else, marking them as a people who had evolved in profound isolation for millennia in that frozen landscape. They were a true ghost population, a community whose physical remains are almost entirely lost to time, yet whose biological legacy was quietly passed on. This ghost did not vanish without a trace. The analysis revealed that this ancient North Siberian lineage contributed significantly to the genetic makeup of the very first peoples to enter the Americas. It also left a subtle signature in the genomes of some populations in northeastern Europe. This means that before the great migrations that shaped continents, there were already epic journeys and meetings happening in the high Arctic, connections between worlds that we can only detect through these faint genetic echoes. The discovery fundamentally changes how we view the ancient world. It proves that the human family tree is not a clean diagram with a few main branches. It is a dense, tangled thicket filled with lost lineages whose stories we may never fully know, except for the fact that they lived, loved, and left a piece of themselves in the descendants who moved on. This is perhaps the most profound lesson of ancient DNA. It has given us the tools to see the ghosts. We now understand that history is not just written by the cultures that built monuments or won battles. It is also written by the quiet, resilient peoples who thrived in the margins of the ancient world, whose only monuments are the genetic sequences they contributed to the future. Every modern genome is a living library, and on its shelves are countless volumes from authors whose names we will never know, their narratives waiting patiently in our cells to be read. Taken together, these 20 threads weave a tapestry far grander than any single revelation. The true lesson of ancient DNA is not found in any one date or percentage, but in the overarching story it forces us to tell. Human history, from the deepest past to the present day, is not a chronicle of purity or isolation. It is an epic poem of mixing, migration, and relentless adaptation. This science acts as a great leveler, democratizing our past. For millennia, history was written by the literate, the victorious, those who built in stone. Countless communities left no written words, their stories presumed lost or silent. Now their bones can speak. Ancient genetics corrects the biases of the archaeological record, giving voice to foraging communities overshadowed by farmers, to nomadic cultures misunderstood by settled empires, and to entire populations that moved through landscapes leaving few physical traces. It reveals a world where the lines we draw are often illusions. The boundary between species blurred when our ancestors met Neanderthals. The separation between continents dissolves in the genetic journey from Siberia to the Americas. The distinction between cultures melts away in the Viking graves holding people from across the known world. This fluidity extends to the very idea of identity. We are not fixed points on a static map of ancestry. Our genetic selves are rivers, constantly fed by tributaries from distant sources, shaped by 1,000 chance encounters and deliberate journeys. Every person alive today is a living mosaic, a unique assemblage of fragments from ghost populations, ancient explorers, and resilient survivors. This realization carries a profound weight for how we see ourselves and each other now. It challenges rigid modern notions of ethnicity and belonging, revealing that the quest for a pure, unchanging lineage is a search for a phantom. Our strength was never in separation, but in connection. The legacy written in our cells is one of endless motion, of meeting, and of finding a way forward together. It is a story that belongs to all of us, a biological testament to a shared and wonderfully tangled human journey. From the Neanderthal DNA in our cells to the ghost populations hidden in our genomes, these 20 discoveries prove that our history is far richer, more interconnected, and more surprising than any textbook could contain. What we have witnessed is the quiet dismantling of a simpler story. The old narratives of isolated peoples and linear progress cannot hold against this genetic evidence. Instead, we see a world in constant, dynamic conversation, where every meeting left a trace, every journey wrote itself into the living code of the next generation. Ancient DNA has moved from a niche science to a foundational pillar of historical understanding. It is no longer just an interesting tool for archaeologists. It is a core way of reading our collective biography. This technology has turned silent bones into detailed life stories, revealing personal kinship, individual health challenges, and profound resilience. It has transformed the grand saga of human migration from a theoretical arrow on a map into a measurable, biological force, with genetic percentages marking the ebb and flow of entire peoples across continents. This science reminds us, in the most personal way possible, that the past is not a static series of events locked away in a museum. It is a living, breathing legacy actively encoded within us all. The food allergies you might have, the way your body fights a common cold, the very color of your hair, could all be whispers from a long-lost cousin or a survivor of a forgotten pandemic. You are a living archive. As the technology advances at a breathtaking pace, who knows what other lost worlds we will recover from a mere speck of tooth enamel or a single grain of sediment. The next decade promises to read even older DNA, to find fainter ghosts, and to connect more dots in the vast mural of our shared origins. If this journey through the echoes in our genes has captivated you, consider subscribing to travel with us into more unraveled mysteries of our past. Let us know in the comments which of these 20 discoveries reshaped your own view of history the most. Was it the intimate meeting with Neanderthals, the lonely resilience on Easter Island, or the quiet departure from Cahokia? Your perspective is now part of this ever-evolving story.